series, and today we're happy to have Dr. Thanh to present for us. He's currently a postdoc jointly between Dr. Jay Zhang, who's the chair of the Biomedical Engineering Department, and myself at the Informatics Institute. He's got many years of experience as a computer scientist working on machine learning and the bioinformatician working on various disease biology problems. For the past 4 years, he's been associated with Informatics Institute, both as a visiting PhD student and a postdoc. Prior to that, he obtained his PhD in computer science from Purdue University. And then he also is one of the person who broke the record from bachelor degree to master and a PhD. I think he got his bachelor and master degrees in 3.5 years and the PhD for another 3.5 years. So he's certainly very gifted and has interesting content to say. Let's welcome Thanh. Go ahead.
All right, so sure. Let me try to, you see my screen and then looks good? Anything to adjust?
Yeah, just put it in slideshow mode and then you'll be good.
Okay. So my talk today is a little more focused on the cardiovascular research. Because this, most of the techniques, machine learning techniques, the new one I am using right now is only being tested in, hard data set. And we are very lucky at BME to have a very good collaboration with, among the faculties here and faculties outside UAB. So we have a lot of support to build up so far with these techniques and to help me to build a machine learning technique, and we test in our data set, which is also unique. So I hope to convey some of my experience and principles rather than copy and pasting code. I don't promise anything about copying and pasting code or sharing library at this point, but I hope to share the principle. So. The overview today, I would spend a bit of time to talk about the characteristic of our data set, the heart, and it has very different natures from other single cell data set that we know more. We often use in the informatics, including the institute, including the stuff I am doing with Dr. Jay Chen in other projects. So goal is different, data is different, and we found that the tools we have been building or have been using, they fail somewhat. They fail to do the results that is needed in the cardiovascular research. So it doesn't mean the technique is wrong, the technique still find, meaningful. Should I say meaningful? I say consistent, consistent results with what we know in the cardiovascular research, but those results does not really help, to address our main questions in cardiovascular research. So this is very important. The main motivation why I and Dr. Chen and our peers at BME department, we decided to move to something else, and we started looking to AI. The first idea was initiated not by me. It was initiated by a friend of mine, Dr. Yuji Nakada. He asked me, do you remember Spark model? Do you know anything like that? And I said, yeah, that is very common in my practice. And then he said, well, because the existing tool doesn't produce the thing we need, so why don't you try the smart model? And from there, I got a few more application in AI, and then and we start seeing the results we need. So coming back to the fundamental points in our field in the cardiovascular research. So the main challenge or the main focus we are having right now is heart regeneration. The heart, very different from many other organs in our body. They don't proliferate well. They are among the least regenerative capacity. So, for example, there's a renewed, the paper I put it in, in New England Journal of Medicine. We talk about, how much the heart cell regenerate or being newly born throughout our human life. And I might say, so from what I remember, we have roughly only 40% of our heart cell being newly regenerated or newly created throughout our life. So that means you might have your heart cell, and all of us, we have the heart cell that exists from the time we were born until we pass away. So that is about the heart. And every year we only see, for normal people, the only 1% or less, cardiomyocyte, which is a lot of the heart cell, being born. So it means, the proliferation, which is the central theme in heart, is very rare. And that is the main issue when, for example, someone got a heart attack, a large amount, usually somewhere between 20% to 40% of the myocyte, cardiomyocyte, was wiped out in an instant. And you can imagine this, it was like a buildup of 20 years. And that is why heart attack or heart injury is very hard to deal with, because simply the speed to regenerate new myocyte is nowhere near to to make up with what we lost in a heart attack. So it is why cardiovascular disease is still the highest cause of mortality in the world today. So, and it is not the only thing. The heart cell, the cardiomyocyte, they also respond to the heart attack or the injury in many ways. Some response is good if they can, proliferate, regenerate. It's good. New cell was born, so the heart kind of gets strong enough. But also, when we have a heart attack, there is a space being open in the heart wall. So the myocyte might grow in size. We call that hypertrophy, simply to fill up the space created by the injury. And this is kind of a temporary solution to fix the injury, to fix the heart. But that is not a good solution, because you don't have new cell. You simply enlarge the cell. A cell become overworked, and in long term that is that becomes a risk for more heart problem in the future. So we don't want that to happen. We don't want our cell to be overworked. But that is a reaction that the myocyte would do if there is not enough number of cells. And of course, when the myocyte could not fill up fully to the space, to fill the space created by the injury, we have scar. And scar in the normal condition is a protection. When you have a skin cut, a small cut on your fingers or on the top of your hand, you might see a scar. This is a protection in normally, but in the heart, this is not good because those protection, those cells being filled or cover, that covers the injury. Those cells don't beat. The heart doesn't beat, and that's basically it's bad. It is not a good thing at all. So, yeah, coming down, Gina, to make the heart cell to be born, it is kind of very challenging. So it's very different from cancer. Cancer, we try to eliminate the cell in the cardiovascular research. We want to make the cell more cell being born. So two different goals. And also the making the cell, making more cell is just one aspect of the heart research. The second main challenge is after we make the cell, making new cell, we must make sure that they function. They need to be so that the heart can remain fully function. So, and talking about physiology, the heart cell is a big cell. So usually human heart cell has 2 nuclei, so that it is big enough to do a contraction. Sometimes it may have 4 nuclei. In peak, 8 nuclei is possible per cell. So because it is too big, so those heart cells, when we try to do single cell sequencing, simply it doesn't pass through the sequencing device. So that is why we in the cardiovascular we do single nuclear. This is the only option. And that means we lost a lot of data that might be helpful about these RNA or other genetic that is not that is outside the nuclei. So what they are, I don't have the experience right now, but that is I can tell. These single nuclei data is somewhat different from the single cell data. Hopefully that affect the analysis quite a bit. And also the heart functionality has metabolic shift, meaning the heart use glycolysis for very fast speed of consuming the energy. That happened for the new cell, young myocyte and other young cell. But then when they got time gone and time passed, and then the cell get more mature, they would switch completely to fatty acid oxidation. So when we do, what it means is when we do a single cell in cardiovascular research, especially when we go with the regeneration, we are very easily to see cluster that is determined by metabolic shift and other physiology. For example, also cell adhesion, cell need to light up to form a wall. Cell need to be it needs calcium and then potassium ion, something like that. And when we do single cell, we are very likely to get the cell cluster associated to those physiology. So, that means the result is not incorrect. It is very consistent with the knowledge we know in the heart, but we don't see cluster that are responsible for proliferation or responsible for hypertrophy, whether we can see the cell are likely to survive, survival gene or something like that, or apoptosis. This is, if we don't do it carefully, we will not find those signals at all. And those signals are what we need, not about metabolic, not about adhesion or contraction. So of course there are, there are certain domain, subgroup who's focused entirely on metabolic cell adhesion contraction, but that is not the goal we have. And so that is why we saw that later I will show the normal pipeline kind of failed to produce what we want in the topic. So a couple of more things about what we want to do in cardiovascular research, especially at DME. So the main direction, there are many ways to approach the heart regeneration. The most promising direction that I might work with is actually single cell and try to make the cell younger, which means we try to find out a population, especially cardiomyocyte, that are regenerative, that can produce more myocyte. So how do we do so? We look at these newborn animals, especially both pig and mouse. So what we know is if the heart, if the animal got a heart attack on the first day of being born, they will fully recover. The heart regenerates, new cell is being born. That is very certain. It has been the main findings for decades. However, when the animal got older, usually after 7 days of being born, and if that animal got a heart attack, it will not recover. It may have a mix of things I just mentioned, hypertrophy, and then new cell may be born, but they don't work anyway, and then scarring. That means they don't really fully regenerate after their injury. And a new finding which Dr. Chen and I was about to publish a paper with the DME is we have a way to extend the regenerative windows. So instead of 7 days and the heart don't recover, now the heart can recover when we do when we do a injury on day 28 of their life. The way to do so is to induce a first injury by cutting the teeth, the bottom teeth of the animal. We call that apical resection or AR1. This just means you cut a little the teeth, the animal will be fine, and then they recover after a month. And on that point, we do a second injury, they would recover. So that means we have a kind of longer time point to examine and find a subpopulation of myocyte that may regenerate. And there are other directions with Yang Zhou in our department. She is focusing on human-induced pluripotent stem cell. So basically, what we test is we take a little tissue from human donors. There are way to turn those tissue to become stem cell. We call those Yamanaka factors. If you knock up those 4 genes, and we have a Nobel Prize for those scientists who found out those 4 genes, you knock up those 4 genes, the normal cell from human becomes stem cell. And they are from those stem cells, there are ways to knock up some other gene, for example, TBX genes. So the stem cell will have the characteristic of a cardiomyocyte. And she did, she knocked up those and she put that into the pig heart, and those human cells survive in pig heart and help the pig recover. And recently we got a heart transplantation, so a pig heart being put into a human body, and that works. The patient, from what I know, after receiving heart transplantation, he survived for more than 3 weeks. I don't know at this moment whether he survived or not. I can ask the person who did that first in the worst experience. So anyhow, Any questions so far to me about the heart physiology and the goal of our research? Okay. If not, then we talked about the pipeline here. So basically, where this is strategy is the same to almost all kinds of single cell projects. So we normalize the data, we find clustering, we try to find any cluster that is meaningful for our projects. In our case, a cluster of heart cell, especially myocyte, that can regenerate, and then we would do pathway ontology, gene set enrichment analysis to show that the cluster we found indeed support the goal we want. For example, in cancer, there are is there any kind of cell population that are more likely to to contribute to metastasis? And in our field, can we show that there are population that exercise cell cycle and proliferate? So, I mean, before coming next, I would one experience I have is usually when we do when we want to find out a subpopulation that proliferate, we would look at the proliferation markers, Ki67, for example, but that doesn't work well in our data for two reasons. Number one, proliferation is really rare, and number two, we are doing single nuclei in pig. The pig genome and is not as comprehensive as the mouse genome, when we can find more, better reference genome for Ki67 and other gene like Aurora B and things like that. So that is more challenging. So that is the first case study I work with to build up my tool in AI. So that is the mouse heart regeneration. First order is Dr. Mel Tui. She's a good friend of mine also, and she gave a talk yesterday at our department, and some of you might attend her talk yesterday. So basically, that is mouse. So she induced the heart injury on the first day when after birth, and then she keep extracting the data. One day after the injury, which means the heart, that means the mouse is 2 days old, P2, 4 days old, P4, 8 days old. So we know that this line, this A there, the heart will fully regenerate. That's right. Yeah. And then she got a control. So that is the heart having injury on the 8th day after birth, and we know, like I showed before, this heart will not recover. So we see these these red lines on the bottom right of the heart that represent the injury area. So this one, almost recovered, and then the bottom one with bigger heart, that looks like recovery. And then she also knows how to use single cell data. She's good with CELRAT. She used CELRAT for the normalization. And then what she told me was she also used CELRAT for clustering, but she has to merge the CELRAT cluster in such a way so that she end up with 5 quote-unquote subpopulation of myocyte, which means there is a lot of ad hoc. And I don't have her code, and that is that means from here we have to do ad hoc. After normalization, you can apply any clustering, but then anyhow we have to do ad hoc. And I try to create my my own replicator pipeline, and I saw so many clusters, and I don't know. I asked her, how do I merge this cluster? And she looked at my data, and then there's no answer from her. I guess one of the reasons is Seurat clustering has good and good, with certain random seed factors. So the results I produce is even cluster number and things like that is not exactly the same to what she produced. Anyhow, and then there are a certain level of ad hoc pathway analysis. When I say ad hoc, that means the way she chose a marker. For example, she lists these 5 markers over here, and then she also validated each marker by immunohistology, knock up and knock down, expansively, NF-YA and ZE2LA. That is her main work in this paper. And I checked these expressions of this one, NF-Y alpha, redoing from our data set and her data set. And the question is, well, this two gene by level expression and p-value is not comparable to many other genes. Why those two genes being big? And that is, ad hoc. So if you attend yesterday talk, she mentioned very clearly about NF-YA. And NF-YA, these genes that mark this number 4, which is the more abundant in the regenerative heart. And then whether this NF-YA is contributing to proliferation, is that population proliferative or have something else? And indeed, and then it turns out she did not focus entirely on regeneration. She focused on cardiac protection, which is NF-YA attractive factor. Still, we see those, especially Ki67 proliferation marker being explicitly expressed in that blue subpopulation. So at least you can conclude that this one is the one contributing to regeneration that will proliferate. Then cell cycle cyclin B1, cell cycle marker, and a few transcription factor she picked out. So she validated all of those. So I choose this as a ground truth to build up my tool. So this is what I my attempt to reconstruct the results using Seurat and scan PY. And number one, the cluster doesn't look clear. So which is fine, I can take care of that. Number two, do I see these CM4 over here? That is explicit for the regenerative heart, meaning it is high in in P1 but low in PH. The answer is yes, we could find such a cluster using both Seurat and scan PY. So you can see here this red one is more like this regenerative CM4 from her results, and this blue one looks like non-regenerative, that is explicit for the PF mouse. We know it doesn't recover. Both sides, both Seurat and scan PY could find such a population, but when we check the expression between of those markers, she clearly validated and found. We don't see all of them being consistent with what she found. So here you can see proliferation marker and cell cycle, especially this guy, proliferation. We don't see the difference all between regenerative and non-regenerative. So simply, that means Seurat and scan PY found out cluster of some other those clusters represent some other physiology instead of proliferation. So, and this is why I try in an AI and I look at autoencoders. So the strategy for autoencoder is that in principle, what it tries to do is to synthesize the cell So we have input layer that represents the real cell after we represented by a vector gene expression by Excel. In the output side, we have exactly the same number of features or genes here. They are all fake, they are not real at all. And then the autoencoder, we try to make sure that these fake guys over here is as similar as possible to the real guy over here. And doing by minimizing the, you can see these X on the left side, the Y on the right side, make sure they are as similar as possible. As subtract them, make it small, minimize that. So this is what the autoencoder does. Of course, there are many layers between this left side, the input, the real cell being processed by many layers over, and then getting to the smallest layer in the middle, we call embedded layer, which is 10 dimensions, then being expanded. Then how many layers? It depends on the computing resources. With what I have, I can afford to do 3 layers. If we do computing with a larger memory and we can spend more time, then we can do more layers. Regardless, the principle here is you take the center layer and you do embedding. Another chat suggests maybe it works because of the data cleaning effect done by autoencoders. I think it is very likely. It's just I haven't validate or verify such an effect by a more well-built experiment. But that is how I did the AI and recluster the cells. Basically, get that, redo UMAP and redo the clustering. And from here, I see, I also reproduce these, that's red cluster right here, more, more enriched, more abundant among these regenerative hard P1, nothing at all or very little in non-regenerative Ph. So I can certainly see specific cluster that represent 2 sides. And I test on her marker again, and now you can see those proliferative markers. They are certainly higher or upregulated in my new, proliferative. My new cell clusters called CM1 here. The other results, the other tool doesn't show that. So that means, the autoencoder find out a subpopulation or reproduce a subpopulation that contribute to regeneration, and those things are validated. So, any question on that? If not, then what do I have to say here? So yeah, I just mentioned it before. So the next question we try to find is, okay, now I can reproduce her results. I can get this red line, the proliferative one with all of those markers. How do I show that this cluster is, this cluster underwent cell cycle and other proliferative processes? So certainly the straightforward answer, straightforward way to answer the question is we take these markers for those red cluster and we do pathway analysis, see if we see cell cycle being enriched or signaling pathway that promotes cell cycles being enriched. And that is a strategy we did for bulk RNA-seq, we did for single cell, and anyway, you take the cluster marker and you do GSEA or David or Pager, or you can try any tool. So, but the principle is the same. Take marker, do sequence enrichment. Unfortunately, none of them works or fully works. For example, I know those 4, the gene ontology, the gene member for those six phases of cell cycles from G01, G1S, that is, I forgot what it is. So GM2, that is mitosis to G1. So basically, I try different tools implemented in CLRAD and other tools, so they don't clearly show that cell cycle are enriched in the CM1 cluster, the rest cluster I just mentioned. So it failed simply. And then those signaling pathway, MARC, HIPPO, CM signaling, JAK-STAT, and RAS, those are what we validated in a formal study in 2020 using MARC-RNA-seq and Western blot. We know that in the regenerative heart, those signaling pathways should upregulate. But in single cell here, we take the standard pipeline, and I don't say the pipeline is running algorithm. It just says it's not sensitive enough to show what we know. So the alternative approach is coming from my first story, sparse model suggested by Yuji Nakata. So basically, what we try to do is, well, now we have the CM1 cell, the regenerative cluster, and the CM2 cell, the non-regenerative cluster. So they are different, black and white. We consider them as a ground truth. So we only choose genes that belong to a specific ontology or pathway. For example, if we want to analyze mitosis, we just choose a mitosis gene to form a data with C1 cell and mitosis gene, CM2 cell, mitosis gene. Try to classify or maximize the difference between these two cells. Y equal to WX plus B. So Y is a score for X cell over here and X cell over here. And we try to make sure that for CM1 cell, Y is as positive as possible. For CM2 cell, Y is as negative as possible. And this is coming back to AI, a very fundamental question we learned from our first year PhD. So, and you can certainly do that with one or two lines of R code or MATLAB code. Now it's easy. And then from here, we have a score for X cells in X cluster, and we can start seeing the difference. So CM1 score higher and in G01, G1S phase, mitosis, and also in other pathways. So certainly those CM1 cell score is higher, and we can do p-value from here. Just now we just become a t-test or a rank sum test comparing two population and get a p-value. And then we, let me give me a minute. I'm in a meeting. I'll talk to you later. Okay. Yeah. Thank you. Sorry for interruption. So basically, with the sparse model, we got all of those, cell cycle phase enriched, and then the signaling pathway, which we know by validation, by independent validation, that being enriched. The other techniques, they may enrich some, but not all. So I can show certainly this is a better way for single cell, at least in our case. I have not tested this guy in other data set yet. For example, cancer, not yet. So, but at least we can tell we got a meaningful result for our cardiac projects. So this is the second case study when we apply directly to our big data. We have a very big data set of 500,000 cells collected throughout the years, and my friend and the good friend Nakada, he's a leading guy in this paper. We will publish soon with Dr. Chen as well. So basically, coming back from the beginning, apical restriction was induced on the first day of the big heart. Basically, just cut off this guy, the heart will recover on day 28. Then the second injury was induced right there, very near to the cutting zone. That is the second injury. And then we wait for another month. On day 56 of the heart, they recover. So, and then we have plenty of samples group, naive heart from day 1, day 28, day 56, and then they are no injury. Then we have heart with apical resection, and then from day 1, day 28, day 56, and then the one with two injury together, the apic AR and MI also. So a lot of data in this study. So we apply autoencoders to, find cell type, and then do a second autoencoder to cluster the myocyte, our focus cardiomyocyte. And then we found a unique cluster. Here we call CM1 that uniquely appear in, in this regenerative group. That is apical resection on day 1 and still appear in day 28. We also find cluster that represent the normal fetal heart, normal heart on day 1 after birth. These orange one, CM7 here, CM8 here. This 9 is, 9 and 6 over here are unique for the normal heart on day 56, which means they already mature enough and no longer, they might regenerate, but with like 1% of rate, like I mentioned. So this talk, this one, directly tells after the injury, heart physiology is no longer the same to the normal heart, no matter how long we let the heart recover. So you see that one here, 7, 8, 9, 6 are unique and exclusive for normal heart. Nothing, we don't see anything of any of them among the injury group, which is one message in the manuscript I just mentioned before. And the good thing of the first cluster, CM1 here, is that it co-up-regulates 4 genes together: TBX, 5, 20, ERBB4, and GRK5 is new. So TBX, 5, 20, ERBB4 are well-known transcription factors that enable the heart to mature and proliferate, especially in the fetals and the neonatal, day 1 after birth heart. So this is not just our results, those are the other papers I cite to show the point, but even in our group, Jancho, she knows this TBX quite well, and her strategy is to use TBX to make new myocyte derived from the stem-like model, the stem-like cells. CRK5 is a new one. I just found one, a few papers. That one is more about hypertrophy and then the reaction in a down heart. So this one, it makes CM1 to be a mix. That is our thinking, and what we might validate in the organoid model. Hopefully, the result will come soon. That this CM1 has both, it preserves the stemness capacity, and it's mixed with some mature heart reaction to injury, and that is one of the interesting things. And we also try to do pathway analysis using the, this sparse model, and then we plot those bar graphs about the percentage of cells that have a high score for muscle cell proliferation and muscle tissue growth. And coming back from those slides, CM8 is fetal, CM7 is day one after birth, so they should have very high score for muscle tissue proliferation, and we reflect it right there. We show it right there, they are high. And then, interestingly, that CM1 I mentioned, the red one I mentioned, also have high capacity for proliferation. So basically, we meet our initial objective in finding a subpopulation of cell in chiromyocyte that can grow and proliferate. And there are a lot of things we go on this year. I hope to tell you interesting things soon. But that today, that is as much as I might tell in the cardiac. There are a lot of results ongoing. So I thank the department chair, Dr. Jay Zhang, a good friend of mine, Judy Nakada. He helped me a lot in explaining those things, and there are many other people here. And then certainly thank Dr. Jake Chen from the Informatics Institute, the two co-advisor helped me a lot in the process. So that is the end of my talk, and I'm very happy to take any questions.
All right. If you have questions, raise your hand so I can start with a question. So you're trained as a machine learning person. You go through bioinformatics training, and then you go to a more biology-oriented lab. Do you feel like having any challenges to go from computer science to this field? And what is your plan to actually continue your skills in computer science, where you're going to actually become a biologist?
This is a great question. That is actually related to the perspective. So what I learned a lot after working with you for a few years and with many other people is the difference between me and other computer scientists is the way we perceive the biology and the problem we are dealing with. I can tell directly, don't look at the biology problem in the same way to many other machine learning, AI, and bioinformatics and like that. And certainly, one way I can tell is when we get deeper in biology, we see new problem. That new problem demands new tools. And now this is a synergy between three things: informatics, biology, and AI. So if we just stop by, that is my experience so far, reusing or redoing what people have done, we don't see new problem. We might improve the tool a little, which is fine. And there are certain people who choose that direction. But I choose, to see more problem. I see new thing in the data or the field that needs new techniques. And of course, we have to be, the perspective has to be very broad. Some people only think from one perspective, which is fine, we are trained that way. So I understand why. And if we have just one perspective, like I just want to deal with computer science, then we don't see more perspective. And then you choose that direction.
I don't see other questions. Well, is your goal to understand how the heart got injured and then the process, the biological process, or is your goal ultimately to find the drugs?
Well, the question, the answer is to find the final goal is to find the drug. But to find the drug, we need to understand the process. So they are the milestone we have to get through.
Okay.
Yeah, and you know, in the heart cell, because they don't proliferate, we cannot culture them. If we can culture the heart cell, we can certainly do screening and find the drug, but that doesn't happen in the field. So,
But, so maybe related to COVID, a lot of people with either vaccine or long haul COVID have pericarditis or myocarditis. I wonder if your finding here could be applied to treating COVID long haul patients.
so first of all, we need to understand how they got heart issues. There is a new faculty coming to our department today. I mean, last week, and he talked to me and he said, the cause of injury makes the cell behave different and makes the treatment process different. Apical resection means you cut the cell. So there is no need to do, healing, because you cut the cell, the other cell still appear there, they still function. So cause of injury different. You don't have to do any immediate step. But when you do myocardial infarction, there are cells dying over there. We have to clean up those cells first. If we don't clean up those cells and their regeneration happening, but that will not effective. It will be meaningless without cleaning up those cells. So that is why we have to know how they got heart issue. Is that because of the cell dying or cell underwent certain apoptotic process, or just because they can get instantly wiped out, things like that, or the cell got larger or smaller, the heart slow in beating, or things like that.
Okay, we have one question from Rahul.
Yeah.
Hi, Dr. Thanh. It was really interesting. I have been talking to you for long, and I am pretty curious. Like, first of all, what is the basic difference between single cell and gene expression or genome?
Okay.
Is there a fundamental difference, and how can we use this single cell to do drug discovery?
So to say difference is an inaccurate word. Genome is a concept. Single cells is another concept. So when you mix these two concepts together, you have single cell genome.
Okay.
It is like apple and red, and you have apple red, right?
Okay, so that's the fundamental difference, because I have been reading a lot. I'm like a computer scientist like you, so I have been going through a lot of turbulence in my head, especially the biology part. So
Yeah.
these may be a very basic question for a biologist, but for me these are important questions, and I need to ask it so that I don't have to find it by myself. And the second question was, how do we use it for drug discovery process, especially for drug screening?
Okay, there are, I think, from what I know, two strategies. Number one, if we can cluster those cells and make the cell grow in the cluster dish in the wet lab, we can treat those cells using any drug, and then do single cell, send them to sequencing, and that is one way. And that happened in cancer, because we can extract these tumor cell. We can grow them, create a cell line representing the tumor, grow them in the room, in the lab, and then treat them, and then send them to sequencing. Second way is, but when you isolate those cells from the human body, many of these physiology change disappear, or something that we don't know. So the thing we deal with in the wet lab is no longer similar to what we know in human inside the patient, right? So certain strategy is you take the stuff directly from patients, send them to sequencing, and then try to find out which population which cell population has the capacity to do what you are looking for. For example, which population can proliferate? And what is the signal, I mean, the signatures of this population, what gene they upregulate there? And we try to, design a drug that help those genes to upregulate. So.
Great. Well, we're over time. So let's thank Dr. Nguyen again for the wonderful talk. We'll see you next time.
Yeah, thank you.
Bye.
Bye.